it's the first lesson of the second week. And basically what we're going to do today is we're going to talk about the scientific method, explain a little bit what it is. So we have seen what is science, so you know now more or less what science is. And to do science, we have a scientific method, and this is what the class of today will be about, okay? I will explain a little bit how this is in Canvas, the theory about this. There will be a video, like a short video that explains a little bit more about the concept. And then we're going to use for the first time the tool which is called Educa Play. So it is integrated in Canvas, and it's really, really nice because if you do this, it's not for points. I mean, you get points, but they don't count. So it's more like a test for yourself. And I would say that it's important that you do this well, because these exercises, they have really been designed to go inside a little bit with the exam questions. So if you know the answers of the Educa Plays, then your quizzes and your exams, you most likely will do them well, because the questions are similar, okay? So that's why it's important. And then to finish the class, I will explain about the elephants in Africa. So you have to do a little homework about that this week. You have to hand it in on Friday, so you can do it today or tomorrow. And that's basically it. So the scientific method. So you can already see my background. The scientific method is something really interesting. I'm not going to explain what you see behind me, but what it really means. I hope that you can do it once we go through the theory, but just have to have a look at it already. You see like a red tree or a red plant and a green plant. And one plant, let me move a little bit. One plant is in the house and it has like a light bulb. It's like in a little shed, and the other one is in open air and it's exposed to the sun, okay? So that's a little bit what it's about. And my idea is that you start naming the variables that are there. So if we go to Canvas, you all have access to this. I'm going to share it, should be shared right now. So let's go over the theory, okay? You click nicely on it. This is what, here you can see where you can click. In case you didn't know, you click on this here, and then it says where you can click. This is the home button, and here you can go to the index again, okay? So if we click on here, if you click on the arrows, you go basically through the whole content, and that's what we would like, okay? So the scientific method, okay? So we have seen what science is. And to do science, we have something what we call the scientific method. There are certain steps in this method. We're going to see that. And the method includes doing experiments, okay? We're going to see the very, very basics of it. And we're going to see why doing experiments is important, okay? Okay, so the scientific method is based on a timeline. Basically, science, we do science to solve problems. We have seen that, you know, we have a lot of problems in the world, and we try to fix them through science. And science, what we have also seen, it's a really good method because it has given us the best results of anything, basically, in medicine, in technology, in transportation, in agriculture. Think about what we discussed last time. There are so many problems, and basically any problem that we face, we can solve through science. So you're going to observe. That's the first thing you do, okay? Observation. You have curiosity about something, or you notice a certain problem, and you're going to say like, okay, why would it be like this? Or how can I solve this problem? You know, that's observe. You think of an explanation. That's the second thing. The explanation has an important word in it, a hypothesis, okay? A hypothesis is basically an explanation, but the big difference is that it's like it says here, it's a testable explanation, okay? It's something that you can check with an experiment. So a hypothesis is basically something that you can verify by doing an experiment, okay? An example, like you can see here, the number of birds is decreasing because of the number of cats is increasing. You can check this because you can count the amount of birds and you can count the amount of cats. Okay, so this is something you can test. Think about what this article was about that I told you last lesson, that in North America, Canada, United States, they did a similar experiment and they noticed that 3 billion birds were missing. Okay, if you take a lapse of 50, 60 years, they discovered that there were 3 billion less birds in North America. Of course, this was not due to cats. What they think, there are different variables here. They think this is due to pesticides, to loss of habitats, deforestation, and monoculture of agriculture. Okay. Then the third step is evaluating the hypothesis. So to go to the example of cats and birds. So if there are more cats, then it's possible that there are less birds. Or if there are less cats, it's possible there are more birds. Okay, that's evaluating. Then you do the experiment. So you make a model, you make a controlled experiment, and you're gonna count. Okay, this we're gonna see more in detail. And deciding is in the end to see if your findings are correct or incorrect. That's basically the conclusion you make. Okay, this is very theoretical, but once we go more into the experiment, it will become more clear. So let's watch this video about a scientist in London that discovered why there was so much cholera in London. Okay, I think it's a very nice video. Cholera, we think about nowadays that it's a very, very tropical disease, but cholera used to be something that was very, very real in Europe. So let's have a look at this video and let's see what the scientists discovered a long time ago already using the scientific method. So basically it's a summary of what I just said, and it's in practice what they did in London. So hopefully it starts, because it should start. Okay, here we go. You can all see the video. I mean, it's not started yet, but it's loading.
Yes.
Okay. And sound is okay, because
the scientific method usually goes something like this.
Yes, it's okay.
Perfect.
Make an observation. Hey, there's a pizza. Form a hypothesis.
I bet that pizza is delicious.
Test your hypothesis.
I'm going to eat this pizza for science.
Record your results.
Hypothesis confirmed.
And boom, science complete. But in reality, science is not a simple linear process. It's a dynamic, interactive process with several key areas that interact with each other. Observations lead to questions and new ideas. Scientists form hypotheses that predict results, test them with experiments or observations, and interpret the results. Scientists discuss their results with colleagues who bring new ideas and questions. Public issues inspire new studies that may support public policy, technical innovations, and new questions. To fully understand the process, let's visit the story of Dr. John Snow. In 1831, thousands of Londoners died in an outbreak of cholera, a terrible diarrheal disease. John Snow had observed the horrors of cholera and had questions about what caused it. The popular belief was that miasma, or bad air, caused this disease. But Snow observed that cholera first showed symptoms in the bowels, not in the lungs. This led to his hypothesis that cholera was spread by drinking contaminated water, not by breathing toxic air. In 1849, Snow published a book that shared his ideas with other scientists. But without proof, his hypothesis was generally ignored. When cholera returned to London in 1852, Snow gathered scientific data to support his hypothesis. He went door to door, mapping cholera victims, then compared his data with sources of drinking water. The most notable outbreak occurred in the Broad Street neighborhood where Snow himself once lived. Snow was able to trace the Broad Street outbreak to a single water pump that had been contaminated by a leaky cesspool. He also noticed there were no cases of cholera in the neighborhood brewery because the employees there didn't drink water, they drank beer. Snow concluded that cholera was caused by a germ that was transmitted by drinking water contaminated with infected sewage. In Broad Street, the pump handle was removed and the outbreak ceased. This was the first positive result of Snow's study. However, the source of contamination, London's leaky cesspools and open sewer system, still remained. Snow then published the results of his research, which clearly supported his germ theory. Unfortunately, public officials still rejected Snow's evidence, and nothing more was done. Coincidentally, just a few years later, construction began on a new sewage system to combat London's foul stench. These improvements to London sanitation services quickly reduced the number of deaths due to cholera and other waterborne diseases. It wasn't until 1883 that another scientist, Robert Koch, first observed the germ responsible for cholera, the bacterium Vibrio cholera, which finally confirmed John Snow's germ theory. The effects of John Snow's pioneering work continue to this day and beyond, all thanks to the wonderful process of science.
Okay. So, there we are again. So let's, I would like to resume a little bit. In this situation, if we see the scientific method like more than 100 years ago, what was the thing that John Snow started with? Who can tell me this? So think about the timeline.
Like he's
An idea.
Yeah. So what was the first thing that he did? So I'll share again quickly. Just to
like he saw the cholera. He saw the cholera and he was like, how can we fix this, or what's causing this?
Okay, very good, Santiago. So the first thing that he did, he observed. He saw there were cholera cases in London. Okay. Then, Emilio, I saw you also trying to say something. Think of an explanation. What was the explanation that John Snow thought of? Give me two explanations, because there was the common belief and what he believed.
Well, the common belief was that it was spread through dirty air, and he thought it was spread through dirty water.
Yes, very good. It didn't make any sense that it would spread through air, because people, they got diarrhea and pain in the belly and not in the lungs. So John Snow thought of an explanation, and he thought it might be, it might have something to do with the water and not with the air, what people were thinking. So who can tell me like a hypothesis that he formulated? So what this is the hypothesis that they formulated? It's yes.
But the dirty water, like the sewer water, was combining with the water sources, like wells and stuff, and that was causing. And there was a bacteria in that dirty water, and that's what was causing the cholera.
Yeah, that's a good hypothesis indeed. Yeah, he said like it could be that the water is contaminated. He didn't really know why it was contaminated, but it was later on that they discovered that it indeed was mixed with sewer water, which is very often the case. Then he did an experiment about this. First he couldn't do the experiment because the thing what he did is he talked to other scientists and he just told his hypothesis, and they didn't buy it from him because there was no proof. So you see, it's important, as John Snow also discovered this, that if you just come with a hypothesis, then nobody is gonna buy it, you know? You have to come with the experiment and you have to come up with the data and the evidence. So it took him almost 10 years, it was from 1840 till 1850 something, that he actually did the experiment. So what was the experiment done, Ana Sofia?
What, sorry?
What was the experiment done that they did in London to make sure that indeed it was the water that was contaminated?
they like made like proofs with the water that was like filtrate, like the thing that give water to that like colony. They made like research and like tests to that water.
No, not really, Anna Sofia, but who knows what they did to make sure that indeed
I
they...
know. They researched the amount of people that die drinking from the water of the, how is it called? The black thing that removes water from the dirt.
Yeah,
from the
it's good, Emilio, indeed. What they did is they were mapping. So Jon Snow went asking people, oh yeah, who is sick, who has cholera? And he put everything on a map. And on the map, he could see like, okay, this area seems to be a lot affected. And then he could pinpoint all these people are drinking from this pump. As a consequence, they removed the lever of the pump, which is an experiment basically, because you don't allow people to use the pump anymore. And what did they see? People didn't get sick anymore. Okay, so that's the experiment, and that came to the conclusion that indeed the water, which is basically the proof that the water was contaminated. Okay, so whenever you do an experiment, you have certain, we call that a controlled experiment. In fact, you have certain variables. Okay, it's something to have cause and effect, and that's why look at my background, because I wanna explain it simple first. So a controlled experiment is that you compare two things with each other. Okay, and one thing you don't manipulate it, and the other, which is the control group, and the other thing you do change something. So if you look behind me, what you see is two plants or two trees, and they have two different colors. Okay, but let's say they're two the same trees. So what is the same in the experiment when you look to my background?
They're both the same object, like they're both the same type of plants?
Yes, they're the same type of plants, but we see that they have a different color, right?
Yeah, it's like a different environment. I would think control one is the one outside, and the one who's adding the variables is the one who's inside, like a house with a light bulb, to see maybe a photosynthesis occurs with photons, or it has to be sun.
Okay, so what if we have variables? Okay, let's assume the plants are the same, but the difference is that we observe that the leaves have a different color, but the plant is basically the same. Okay, so we have two types of variables. Okay, we have independent variable and dependent variable. And the independent variable is the variable that we manipulate. So that's what we change in an experiment. Okay, so if you look behind my head, you see two conditions, and there is one variable that is different in both situations. Okay, then there is the dependent variable, which is basically the same. Okay, so who can tell me what is a manipulated variable in the background you see behind me? So I have two plants. The plants, they are genetically the same, but they have a different color. They end up with a different color of leaves, of foliage. Okay, but I give them the same amount of water, but like Emilio correctly says, they are in different conditions. So what is the independent variable, which we call, like you can see here, the manipulated variable?
The light bulb?
Yes, exactly. Not really the light bulb, but the amount of light or the type of light. Okay, that's the manipulated variable, because this is causing something different. Okay, so what is the dependent variable here? Is what we measure. What do you measure? What is the difference that you see? So this is causing the effect. What is the effect? What's the color? The color. So, I mean, I don't like the image that much, because usually one grows, the difference you would normally see is that one grows more than the other one. Here they did a different color, but it's the concept is the same. But indeed, that's the dependent variable, okay? So the manipulated variable is the one that is different in both situations. So here, the red plant gets sunlight, the green plant gets electric light. This is what is manipulated, okay? Independent variable, okay? It's called independent variable because it's we cannot control it, and it's causing the cause, okay? And then the effect, which is the dependent variable, is what you see. What is the difference you see? Green and red, okay? Everybody understands this? Because the independent variable causes change in the dependent variable, okay? So if we think about the cats and the birds, what would be the independent variable?
The amount of cats.
The cats.
Yeah, the amount of cats indeed. Because the less cats there are, it's causing an effect in the amount of birds, which is the dependent variable, okay? It's really not that hard. Don't worry if you don't get it yet, we're gonna do exercises about it, okay? So here I give another example. Okay, so here, experimental group, control group, okay? So independent variable is what we manipulate. One gets water, the other does not get water, okay? What is the dependent variable is when we see what the outcome is. We are gonna measure. If we give water, we see that 9 out of 10 seeds grow. If we don't give water, we see that 0 out of 10 seeds grow. So this is a very simple experiment, you know. I mean, we have an experimental group, okay? We have the control group that we don't touch to see. Why do we have a control group? It's to see that the independent variable is indeed a variable. So the control group is the thing where you don't affect, where you don't do the change with the independent variable. So experimental group, you're going to give them amount of water. So the independent variable is the one that you manipulate because it's the one that is in your hypothesis that you believe that is causing the change, okay? So if we go to Jon Snow, the independent variable, what would it be?
The one being modified, like adding water.
Yeah. Okay. So Emilio Bichara, what is it exactly? If you take the experiment in London, what is the manipulated variable?
The people getting sick.
No, people getting sick is the dependent variable because
I know. Removing the how is it called? The la llave.
No. This is the change that you do. The independent variable is what you manipulate. So how you do it, it's what you say. You remove the lever. But basically, what is Jon Snow doing is the thing that you manipulate is who is drinking the water and who is not drinking the water. Okay, and that's the independent variable. Who is drinking contaminated water? Who is not drinking contaminated water? What is the dependent variable? Is looking who gets sick and who doesn't get sick. Okay, it's that simple. Okay, is it clear for everybody? If not, ask me. It's important that you get this. Okay, now it's up to you. So basically you do an experiment. Data is basically you're gonna give contaminated water to 10 people or 20 people or 100 people and you have to write down the results, okay? So that's basically what science is. That's doing the experiment, it's making observance, is doing the experiment and writing down your data correctly. That's why it's so important, guys, also to be punctual, to hand in your homework on time, to when you give a homework, to write your name, matricula, everything, all the details matter. Because if you're gonna do science, it's all in the data. Because if you manipulate the data, you can prove things that are not true. So you have to be really, really important with the data. I have to go a bit faster because I want you to start doing the exercise. This is a video about debunk the junk. I would like to see it. I think we're going to see it because it's important. Because a lot of science is fake science, okay? And I think it's important that you notice what the difference is. After this video, we can go to the Educa Play and you can see if you understand the concepts that we have seen today. So what are the keywords of today is high observation, hypothesis, experiment. The experiment has independent and dependent variables, and then you, out of the measurements of the experiments, you get data. Out of the results of the data, you're going to make conclusions. Okay, so let's have a look at this, because nowadays there is so much fake news, there is so much information everywhere, and people tend to believe whatever they want or whatever is convenient for them. And it's important that you can say, no, this is not true because of this and this and this reason, you know, that you have good critical thinking. So let's have a look at this. In fact, I went to Telmex to get more megabytes of internet, because I noticed that my videos, they charged really slow. And I noticed there is a small difference now, so it's good. For 100 pesos more.
Yes, it's better.
Have you ever wanted to lose weight but
Mejor.
suckers? Well, Dr. Chaz's miracle pills are scientifically proven to help you lose weight without dieting or exercise. Call now and I guarantee.
Wait, before you make that call, stop and think. Is this claim true? Consider the case of a green coffee extract miracle pill that earned advertisers tens of millions of dollars when they claimed it would cause amazing weight loss with no effort. A spokesperson for Pure Green Coffee brand appeared on national TV programs wearing a lab coat and calling himself a doctor of nutrition. Actually, he was an advertising executive for companies that set up fake news websites with names of fictitious news organizations and logos from and MSNBC. Unfortunately, people who purchased the pills wasted their money and didn't get the results they expected. The Federal Trade Commission, or FTC, convicted these companies of false advertising and forced them to return millions of dollars in ill-gotten monies. So how do false claims like these get started in the first place? With a fake scientific study. A different company, Applied Food Sciences, paid for a so-called scientific study to promote their own brand of green coffee extract, GCA. While other studies showed modest weight loss with green coffee extracts, suddenly it seemed that people who used GCA brand lost an astounding amount of fat and weight by doing absolutely nothing. This study was published in a respected peer-reviewed journal. However, when other scientists reviewed the study, they found that the data had been faked. This study was exposed as junk science, and the publication was retracted. The FTC convicted Applied Food Sciences of using false representation for ill-gotten gains. Any scientific claim that seems too good to be true probably is. To debunk the junk, it's important to question such claims and evaluate them with a critical eye. So how can you determine if any scientific claim really measures up to good science? This is how we evaluated the miracle pill claim. First, we check the source. Who paid for this study? In this case, the fake GCA study was funded by the sellers of GCA. Funding by anyone with a financial interest is a red warning flag. Next, we look for bias and hidden agendas. Who profits from false advertising? The sellers of Pure Green Coffee profited from fake media websites and endorsements, all based on the fake GCA study. Any bias in endorsements is a second red flag. Next, we analyze the research methods. Was the research valid? The GCA study altered weights and other key data, so the study was fake. Bad research is our third red flag. The next step is to investigate independent research. Have other researchers reproduced the results of this study? Since the GCA results were faked, there could be no independent confirmation. Our fourth red flag. Finally, we make the call. Are the miracle pill claims good science or junk science? Having failed in every category of our claim evaluation, we call this junk science. You too can use the power of claim to avoid being ripped off, protect your health, and increase your understanding of the world around you. For example, are claims about new technology or health products or climate change true? Before you decide, stop, think, and ask questions. Investigating a scientific claim gives you the power to debunk the junk.
So this is very true, guys. Please do this because I mean, Facebook, Instagram, internet, we get so much information and so much is fake. And even WhatsApp, if you see what people share and cause panic and make people afraid. I mean, I know people that who believe everything that you send them. It's really tragic. Use this method, which we call claim, that you go through the different passes to check if something is true or not true. Okay, and always think about this. You know, who benefits, why would it be wrong, and what's behind it. Okay. For me, it's very important, especially as a scientist, because if you want to be a good scientist and you share fake information, then you're losing credibility yourself, and that's always bad. Okay. In the video, I still have a doubt. Did you hear well? Because I don't know if I have to silence my microphone or not. I silenced it in the middle a little bit, but can you please tell me, because this is something I'm not sure.
Yes, we hear. I heard.
The other was okay.
So I don't have. If I silence my microphone, you still hear the video or not?
Yes, we hear the video.
Okay, perfect. So now we go to the Educa Play. Here it is. Go in your canvas, and there you can find it in the modules, basically in the scientific method, what I've been sharing all the time, because now it's up to you. Okay, do this well, guys. Seriously, if you understand this, if you do this well, then you're fine. There's 5 exercises. You don't have to do the 5 of them, at least not now. I would like you to do 2 exercises, and I like particularly number 1 is nice. Number 2, I don't like it too much because it mixes Spanish with English, but it's okay, you can do it. But I would recommend you now to try number 1 and number 5, okay? So here we have a text, and it's basically a summary of everything we did today. So we should be able to fill in these things, these words into the text. Okay, can you try to do that for yourself?
Yes, sir.
Yes.
So number 2 and number 5, or which ones?
Sorry, number 1 and number 3, I would recommend you to do now.
Okay.
But do it on your own. But I'm here, you know, I'm here to help out, but I want to see, I mean, I'm gonna do it wrong now, okay? I'm just gonna fill in, it's not working. Okay. I'm just taking like this. It's wrong. I'm just filling in it directly. So you click on it and you say the word that is there. This is 100% wrong, but then if you click check, you can see how much you have, of course. I did it all wrong because otherwise you already have the answers. Okay, you check and then you have the correct ones, and you can do it as many times as you want. You just click reboot, you start again, and there you are. So do this, please. In the meanwhile, I would like to hear if somebody has any questions about this. I quickly gonna repeat, because I think what you see behind me is important, because it's the most simple setup of an experiment that you see. Okay, so you have here the red plant is the control group. The green plant is your experiment. Okay, you always need a control group, because if you cannot compare, you cannot have the data. Okay, you don't have proof. Okay, so in my experiment, a normal plant exposed to sunlight, and here a similar plant in a little house with a light bulb. So the independent variable here is what we manipulate. What we manipulate is the light source. So in my control experiment, it's a normal situation. My plant gets sunlight, and what I manipulate is I put the plant in a little house and it gets a light bulb. Okay. So what I measure is what I see. So this is my dependent variable. So what is the difference? My leaves change red. Are red, and when I have it in the little house, they're green. That's the dependent variable is the color of my leaves.
Where do we check this activity?
It's in Canvas, and you go. You go to modules, and then you go to the scientific method content, and then if you click through the end, you come to the Educa Play. So if you're, I'm not sharing, right? So here, were you able to find this? So you just click through it, and then in the end, you arrive to the Educa Play. And the Educa Play is basically to check if you understand. It's very little theory, but it's terminology that you have to understand. But we will work all week with this. So and then here you get to the exercises.
Yeah, I got it. Thanks.
Okay, perfect. So that's exercise 3. If you look exercise 1, sorry. If we look at exercise 3, I always would recommend to make it full screen because otherwise you don't see it really well. Here it's very simple. What you get is you have to, it's multiple choice questions. So there is like an experiment and you have to say light, color, and number of moths attached are examples of what, and you tell me what it is. It starts very simple. Full screen. So you click on it. And then you click next. The nice thing is also what I've seen, if you log in, it saves your grade. And then if you go, you can see the best scores ever of other students of tech. So if you're very competitive, you can log in, and then it saves your trial. It's not for points, it would never count, but it's maybe fun to do. For those who have interest, I quickly wanted to share with you the World Health Rankings of diarrheal diseases. In fact, diarrheal diseases, they include cholera, which is the example of Jon Snow. Of course, in Europe, it's very, very little. We have a little bit in Germany still, but it's not too much. It's not only cholera, it's also other bacterial infections. But you see here, especially in Africa, it's all red. The death rate is very, very high for diarrheal diseases because there is just no sanitation. I've been in Ethiopia, and honestly, it's terrible because there is no drainage. And if you have a village, even a small village all the feces and all the toilets, they just go to the local river, and that's where they get the water to cook, to eat. So this is really a tragical thing, and this is very easily fixed by having good sanitation. In fact, Bill Gates is working a lot in this, in having toilets in Africa that do not dry toilets. Let's say they're trying to develop a kind of dry toilet so that more people can live safely in Africa, because you see, especially there, it's really bad. Also, India, and here Southeast Asia is not so good because of the humidity. So here it's a little bit better because it's a lot of desert. So the drier it is, the more difficult it is for the bacteria to survive. But you see, from the moment that it's more wet, that especially in the equator countries, the situation is still really bad. Latin America is doing a bit better, but Africa still has a lot of problems. You should see the video I sent you in WhatsApp, why about the continents, that why is Africa doing so bad, you know, because there is a lot of ideas about Africans. There's even people thinking that it's an ethnical problem, that they think, or ethnical or race problem, that Africa is still underdeveloped because they believe that that it's a race issue, but it has nothing to do with that. It's way more complex, and I tend to believe a little bit what is this Atlas Pro video about the different climate zones, that it has a lot to do with it, and the times of colonization that also was not really well for Africa. Okay, so guys, time is almost finished. What you have to do, quickly gonna share screen because it's pre-designed. The homework of today is basically the same what you're doing now. It's putting in practice the concepts of the scientific method that we have seen. So here we have the great elephant census. It's a nice video. It talks about how the amount of elephants are decreasing in Africa. And if you download here the document, it's a Word document, you can download it. And basically you watch the video and you answer the questions. I think you can see it here.
Papi, podemos ver pico cebolla en la tele?
Ahorita, mi amor. And you have to give some definitions here and answer the questions of the video. It's not too long, but I think it's, yeah, that was my son. Do you know the song Pica Cebolla?
Nope.
Don't listen to it because it's it sticks in your head for a long time, and he loves it, and I've heard it like 200 times. I cannot cannot listen to it anymore. So you watch the video, you answer the questions, you put the references in case you have, and you check here the rubric like we always do. I also wanted to tell you, certain of you that I don't know if it's of this group, but somebody said like, oh, I'm sorry, my Canvas, I didn't know how it worked. I'm too late with my activity of last week. Okay, I will revise anything of last week that you hand in late. Of course, you don't get the same grade as somebody who handed it on time, but this will be the only time. Okay, you know several days before when you have to hand in something. And I think it's important that you learn to be on time. Okay, so try to always check your Canvas well, and then we'll be fine. Any questions about this great elephant census? It's a nice video. Huh? It really teaches you, and the same time we're talking about the scientific method. You're learning things from England. I mean, from the Jon Snow video. You're learning things from Africa with the elephant census. And I really hope to maintain this the whole semester, that we take examples a little bit from all over the world. So little by little, you get a better understanding of the world. And that's the nice thing about multi, you know, that in why learn biology if we can learn about the world at the same time. So that makes it really nice. If there are no more questions, you can disconnect. If not, I'll stay here for the last minutes.
Bye, mister.
Thank you.
Bye, mister.
Bye, teacher.
Love you.
See you, teacher.